Hi everyone, and welcome to our video all about the light dependent stage of photosynthesis. This is again one of our photosynthesis module series videos, and what we're going to be doing in this video is having a look at the spec reference 5.2.1d, which obviously is all about the light dependent stage. First thing, before we can really dive into the nitty-gritty of this part of the process, we need to know a very quick overview so we get an idea of what we're really talking about. So first of all, when we're looking at the light dependent stage, this is going to be taking place in the grana of the chloroplasts. So that location is important. Make sure you remember light dependent grana of the chloroplasts. And it is going to involve those photosystems we've looked at in the previous video and light energy. And this is a direct involvement of the light energy. What we're going to do then is have a look at the four parts to this process. The first part, absorption of light by those pigments in those light harvesting complexes of photosystem 1 and photosystem 2. Part 2, photolysis of water or photolysis of water, depending on who's saying the word. Then we've got photophosphorylation, and finally the formation of reduced NADP. So that's our overview of the four parts. What we're going to do is then delve into those in a bit more detail so we understand exactly what happens at each stage. Now, if you remember, when we were talking about this whole light harvesting and we were looking at the idea of the different photosynthetic pigments in the previous video, we mentioned two types of photosystem, photosystem 1 and photosystem 2. Now, what we actually find is photosystem 1, which we can nicely abbreviate down to just PS1. This is found on the outer surface of the grana and the intergranal lamellae. So if you remember, our grana were those little stacks of the thylakoids. My diagram is amazing. You can see why I normally just grab one from the internet. And then we've got those intergranal lamellae, which are obviously those little bridges almost between those stacks of our thylakoids, the grana. So that's what we're talking about. On the outer surface of those structures, we find our photosystem 1. And at the primary reaction center, because remember this basically is like a little funnel, then down at the bottom here we have our primary reaction center, and this has got our chlorophyll A with the absorption peak of 700 nanometers. So that is our P700 down there. We then also have photosystem 2, PS2 for short, not the games console. And what we do is we find this one on the inner membranes of those granule stacks. So remember, we talked about our chloroplast, we talked about obviously different membranes, etc. Then we find our photosystem 2 on the inner membranes of those granule stacks. Now, the primary reaction center is still chlorophyll A, but this time it's got that absorption peak of 680 nanometers. So it's the P680 that we find on our photosystem 2. When we're talking about this process of light harvesting then, what we actually find is that because we've got that array of different pigments present within the actual chloroplasts themselves, then we can absorb many different wavelengths of light. Because as we saw previously, those different pigments have different absorption frequencies. Now, what we find is at the point that the light is being absorbed, and light, remember, is a form of energy, then the electrons are going to become excited. So what happens once those electrons are excited? They actually get enough energy to leave the chlorophyll altogether. And this is going to be really important as we go through this process. So the next stage then is that we are coming on to see what happens in terms of water. In our photosystem 2, we have an enzyme that is going to split water when light is present, because that light is going to provide the energy required for this process. Now, what we have is a process called photolysis or photolysis. Now, if we break that up, we've got two parts to the word. Photo, which obviously refers to light, and lysis is all to do with splitting. So what we're actually doing here is we're splitting using the energy from light. Hence, we split water when light is present. what that looks like then, we start off with our water molecule, and then we're going to generate our hydrogen ions, electrons, and oxygen. Now, because we need to make the full oxygen to balance this, then we would need 2 water molecules. We then have 4 protons or hydrogen ions, 4 electrons, and 1 molecule of oxygen O2. That is, if we think back to the very basics of photosynthesis that we were taught at GCSE and below, when we look at photosynthesis, we always said it is water plus carbon dioxide making our glucose and oxygen. So obviously, when we have a little look to see what we've got here, there's the water. So we've got our water going in and we've got oxygen coming out. So we've talked about a couple of parts here. Now, the oxygen that we produce as a result of this photolysis then is going to be used either in aerobic respiration, or we can release that to the atmosphere by diffusion through the stomata, those small pores on the leaves. In terms of why this photolysis is important, it's going to produce oxygen. That's kind of one important thing on its own. It also provides us with 2 other important parts. Those protons, which remember are the hydrogen ions, are then going to enter this next stage of photophosphorylation. The electrons, which are our little E-, these are going to be donated to the chlorophyll to replace the ones that were lost when the light was harvested. So remember what we said is that those electrons were getting so excited that they were leaving the chlorophyll. You can't just constantly lose the electrons. They need to go back sometime. And this is where they are. Okay, these electrons here that we get from splitting the water are then going back into the chlorophyll to replace the lost ones. Our third stage then is photophosphorylation. First thing, what on earth does that delightful word actually mean? So what we're going to be doing is generating ATP from ADP and our inorganic phosphate. So that bit is familiar, ATP formed from ADP and inorganic phosphate, but it's going to be happening in the presence of light, hence the photo bit in front. Photo meaning light. Phosphorylation, we're adding phosphate, in this case to ADP to form the ATP. Now, we have 2 types of photophosphorylation that we need to know about. Non-cyclic photophosphorylation, and that one involves both photosystem 1 and photosystem 2, or cyclic photophosphorylation, which is only photosystem 1. So what we need to make sure of then is that we are aware of the 2 types and, bad news, what happens in each of them. So this is where I suggest you get a nice piece of paper set out to just lay it out nice and clearly to understand each one in turn, so you don't mix them up. We're going to go through the non-cyclic photophosphorylation first. So what we're going to do, take a little bit at a time, explain what's happening with my stolen from the internet diagrams. So first thing that we've got then is, if we have a little look at the picture on the left, we can see we've got our thylakoid membrane. So we've got obviously the membrane structure there, the bilayer, as we can see, and then we've got that structure of our photosystem embedded within. Now we've got photosystem 2 here. This is the one we're going to focus on first of all. How do we know it's photosystem 2? Because that reaction center has P680 in it. So what happens? Our light in the form of this photon is going to strike our second photosystem, photosystem 2, and what happens is it then travels down through these little substages, which you don't need to know the details of, until it reaches the P680, the primary reaction center. When it hits the P680, it's actually going to excite a pair of electrons. Now, those electrons are going to be so excited that they're going to escape the chlorophyll and they get passed into the thing called an electron carrier in our thylakoid membrane. So what we've got in this very first step, light energy is going to travel through our photosystem 2 complex until it reaches our primary reaction center. 2 electrons are excited, they escape the chlorophyll, and they're passed to an electron carrier in the thylakoid membrane. We will mention here the fact that where we've got our photolysis, or photolysis of water, then we are generating, remember, those 2 electrons, and they just feed back into our P680. So you can see at the bottom, there's our water, this is where it's being split, and the 2 electrons are then replacing the ones that just got excited and left. Now, at the centre of this electron carrier, we have an iron ion. When those electrons are gained, what we see is a reduction. So remember, we're going back to good old oil rig. Oxidation is the loss of electrons, reduction is the gain. So because the iron ion has gained electrons, it is reduced, and therefore we find it becomes Fe2+. Then that electron is going to be donated to the next one, because we've got a whole chain of these electron carriers. And then as it basically passes that electron to the next carrier in the chain, that first one becomes reoxidized. So that means it goes back to Fe3+. This is then just going to continue going one electron to the next electron carrier and so on, all the way down this thing we call the electron transport chain. So basically we're just transferring those electrons down the chain. Why, you might ask? What is the point of doing this other than just sharing electrons around? Well, quite simply, at each of those little stages, as we're going down the electron transport chain, some of the energy from the electron is being released. Now, that energy is actually being used to pump protons across the thylakoid membrane into the thylakoid space. So what we're seeing is, as the electrons are going down this electron transport chain, they're giving up some of their energy, which is then being used to pump hydrogen ions into the thylakoid space. This then leads to an accumulation of hydrogen ions, and we then set up a proton gradient across the membrane because we've got a higher concentration on one side than the other. This then means that we are going to have a little situation where the protons basically want to cross. Now, eventually those little electrons passing down their chain are going to be captured by our chlorophyll A in photosystem 1, because just like we saw previously with photosystem 2, we've had the light energy absorbed, it's passed down to the photosystem, electrons have been excited and left. So we're replacing them at the end of the electron transport chain in the chlorophyll of photosystem 1. We then have those electrons that have been lost being passed into this stuff called ferredoxin. Now that is just a protein ion sulfur complex that is able to take the electrons from photosystem 1 and passes them to the NADP that we find in the stroma. Now, what we then find is that these protons, with their proton gradient, remember, are going to then just diffuse down the proton gradient through a special channel. And the key thing about this special channel is it's associated with an enzyme called ATP synthase. We know it's an enzyme because it ends in ASE. Now, ATP synthase, have a look at the first part of the name, ATP synth, it's going to be used to synthesize ATP. So as those protons move through those channels associated with the ATP synthase, then they're going to trigger the formation of ATP from the ADP and inorganic phosphate. Those protons are going to be accepted with electrons by the NADP. That then reduces NADP, and that is a reaction we're catalyzing with another reaction, NADP reductase. So one thing to note here is when we are writing our reduced NADP, please remember it's NADP first of all when we're talking about our light dependent stage of photosynthesis. And if you want to write it, obviously you can do the NADP with the word reduced in front. You don't have to worry about putting hydrogen ions on because quite often people mess it up. Not going to lie. The far easier way to get it is just write reduced NADP. Let's not risk putting the wrong hydrogen ions in the wrong places here. So here's our lovely overview all together now. Here we've got our photosystem 2, absorbs the photon of light, electrons then become excited in the P680, transferred into the electron transport chain. As they go down the electron transport chain, some of the energy is being transferred to pump hydrogen ions across into the thylakoid space. We also, at the same time, have photosystem 1. There we have the excited electrons leaving P700 and then going through the ferredoxin. We are then going to have our hydrogen ions being able to cross through our ATP synthase associated channel to generate the ATP and of course to obviously utilise the hydrogen ions to form our reduced NADP. We now need to understand the second process of photophosphorylation, cyclic photophosphorylation. The good news, this is easier. We only have one photosystem involved, photosystem 1. Light strikes photosystem 1, pair of electrons become excited and they leave our chlorophyll. They then enter this electron transport system, and as a result of being passed through those different electron carriers, then we generate a small amount of ATP. Then those electrons go back into the chlorophyll of our photosystem 1, hence cyclic, because what we see is the chlorophyll loses electrons, it goes down these little electron carriers before it goes back to where it started. Now, we have no photolysis or photolysis occurring, therefore no oxygen or reduced NADP is generated. So in our cyclic photophosphorylation, all we're generating is a small amount of ATP. I always find it's quite useful at this point to have a comparison between our two types of photophosphorylation, because people do mix them up, and just having a simple table like this is going to help you to get some of those key facts straight in your head before you try to go through the detail of the non-cyclic photophosphorylation properly. So what we've got first of all, the difference in the photosystems. Cyclic, only photosystem 1, non-cyclic, photosystem 1 and 2. Because obviously the different photosystems, we have different reaction centres. So P700 in our photosystem 1, P680 in our photosystem 2 there. Cyclic, as the name suggests, electrons are cycled back to where they began, whereas the non-cyclic, the electrons do not cycle back. They end up in a different place to where they started. Cyclic does not have any photolysis or photolysis of water, whereas the non-cyclic does. We generate ATP in both, but in our non-cyclic we also get reduced NADP or NADPH. We do not generate oxygen in cyclic, but we do in non-cyclic. So I'd suggest that what you do is get those key facts sorted in your head first of all, and then go on to actually get those individual stages, particularly of the non-cyclic photophosphorylation, straight in your mind so you can explain what is happening at those different parts. As always, I do suggest that you subscribe to the channel so you can see when I next upload a video, and of course head on over to the A-Level Biology website to see some other resources and bits and pieces that will help you with your A-Level biology studies.